Evolution's creative solutions, 2 sources of protein diversity. We would like to tell you about 2 different examples of biological innovation that have contributed to the incredible variety of proteins, gene duplication and alternative splicing.
Proteins are really important biological molecules. They're deemed the workhorses of the cell because they're involved in almost every biological process. They have a diverse set of functions and orchestrate almost every part of the cellular symphony. The central dogma of molecular biology refers to the process by which genes become proteins. These instructions are written in the form of genes encoded by DNA. A protein is produced by machinery in the cell that interprets these instructions for creating the protein. Double-stranded DNA is transcribed into single-stranded mRNA, which is ultimately translated into protein. What drives protein diversity? One source of protein diversity is gene duplication. This random event can allow for one of the copies to undergo mutations without jeopardy if the function of the original gene is preserved in the other. Over time, these mutations can culminate into something that performs a new function. If it improves cell function, natural selection takes over and retains those changes. We'll talk about 2 different types of gene duplication. One is tandem gene duplication, which happens during the crossing over phase of meiosis. Meiosis is the kind of cell division that produces gametes, which are sex cells such as egg and sperm. Crossing over is the step in meiosis during which aligned homologous chromosomes exchange a portion of their DNA. Sometimes chromosomes don't align perfectly. They break at different places, and the gene ends up duplicated on one chromosome and absent on the other. Whole genome duplication occurs when chromosomes fail to separate during meiosis. In humans, the gametes that result from meiosis with non-disjunction of chromosome 21 in the production of eggs can combine with a gamete that results from normal meiosis, a sperm shown here, to produce a zygote with extra genetic material that ultimately results in Down syndrome. In both examples, the duplicate gene, if it is conserved, creates genetic redundancy, as both duplicates could carry out the same function. One copy is sufficient to carry out its usual role in an organism, and the redundant duplicate is tolerant of mutations that may change its expression pattern or function. Ultimately, the duplicate genes can either split the work that the ancestral copy used to perform, acquire a completely different function, or simply not be perpetuated. An important example of gene duplication lies in blood clotting enzymes. There are more than a dozen of them in the blood clotting cascade, which helps patch up a damaged vessel and stop bleeding.
Yet another source of biological innovation is alternative splicing. Alternative splicing is a differential inclusion of exons when pre-messenger RNAs are being modified before they can be translated into proteins. So, what exactly does alternative splicing accomplish? By including and excising different exons, 2 distinct mature RNAs can be transcribed from a single gene. Because one gene can make different mRNAs, the final protein products will be different as well, both in structure and in function. For example, the 2 protein products could both be receptors, like the proteins shown here. The differences in their structures would mean that they have a lock and key fit for different particles. So, the receptors would influence the cell in different ways. In fact, some genes are so heavily spliced that they can give rise to thousands of different proteins. For example, the Drosophila gene DSCAM has over 38,000 RNA isoforms that produce over 38,000 distinct proteins, exceeding the total number of genes in its entire genome. The vast proteome that DSCAM gives rise to results from only 4 variable exon clusters that are alternatively spliced. Each different protein is then displayed on the cell surface of a single neuron. The unique protein tag on each neuron can only bind with protein tags of neurons the cell is supposed to make connections with. A self-avoidance mechanism is developed, and only with a splicing-mediated mechanism can Drosophila have normal neuronal circuit development.
As you can see, gene duplication and alternative splicing have profoundly contributed to the diversity of proteins we see today. Gene duplication creates fresh genetic material and allows innovative mutations. Alternative splicing is a biological shuffling that recycles and reinterprets the existing genetic code. In both cases, the forces that drive evolution have greater creative ground on which to act. The resulting masterpiece is the incredible variety of proteins in and around us.